We are coming to the conclusion of our two-day live extravaganza with a few people on Zoom. Actually, not a few people, it's quite a few people on Zoom. I am here to introduce our keynote speaker, Dr. Gina Poe. So I'll tell you just a little bit of background. If I tried to go through Gina's CV, we would be here all day. So I'm not going to do that. I'm just going to be brief and say a few things just by way of background. So Dr. Gina Poe, as many of you know, is a SoCal native. She got her bachelor's degree from Stanford University and her PhD from UCLA, working with Ron Harper, who's a giant in sleep research. Her thesis research was on the neural mechanisms of sleep states in cats. So if you're curious about cats, you can talk to Dr. Poe about that. Then she completed her postdoctoral fellowship at the University of Arizona with Carol Barnes, also collaborating with Bruce McNaughton and Jim Knierim. And there she did some really incredible, exciting work recording hippocampal place cells and looking, of course, at the importance of sleep in memory consolidation. And in particular, the role of REM sleep and the idea that REM sleep allows you to restructure memories and selectively strengthen newer memories and maybe de-strengthen or weaken older memories. Of course, many of you will also recognize her pioneering work in space. She talked about a little bit yesterday. So during that time, she tried to understand the effect of weightlessness in space on place cell coding and what that does to spatial maps. So that was part of the 1998 Space Shuttle mission. Again, something that she shared a little bit about last night. And we learned why she couldn't go up in space herself, but which I'm sure was disappointing, but we still, hey, got rats in space, so why not? After postdoc, she began her career as a faculty member in 1998, first at Washington State University, and then moved to University of Michigan in 2001, and then joined UCLA as a full professor in 2016. Her research lab, of course, focuses on how sleep supports learning and memory, but also, I think in more recent years, you've also started to look at applications to disease, Alzheimer's disease, schizophrenia, PTSD. So very, very exciting. Now, aside from the exciting research that you hear about today, I want to mention another angle to what Dr. Poe does. She has, of course, mentored countless students at every level and has been actively involved in mentoring programs for a very long time. But her efforts in diversifying the neuroscience workforce are truly a model to be admired and to be followed. So, and there's no shortage of involvements in that arena. At UCLA, she's director of the Mark U-Star program, the BRI SURE program, and the Compass Life Sciences program. These are all programs that are focusing on increasing representation and ensuring that individuals from underrepresented groups do participate fully in the biomedical research workforce, and in particular in neuroscience. Nationally, she's, of course, director of the MBL Spines course, which is phenomenal. You should look up the Spines course if you don't know about it. And also co-directs the Society for Neuroscience's highly coveted and incredibly popular NSP program, the Neuroscience Scholars Program. And I actually just learned a couple of days ago that that program got the nation's highest mentoring honor in 2018. So it's a phenomenal program. How many of you know about NSP or have been part of NSP? Yeah, it's a phenomenal program. Thanks to Gina. Now, all of this, you know, this is kind of the technical things, but on a personal note, I just want to say over the period of time that I've known Dr. Poe, I've been nothing short of inspired and truly just humbled by everything that she has done. And I take my cues from her. And I'm not just now starting to get a little bit more into some of these diversity, equity, and inclusion efforts. And everywhere I look, it's Gina. So I'm learning from the best, which is really, truly special. And one of the things that I just want to say that, you know, in addition to, of course, the incredible quality of science, her personal energy and commitment to mentoring and commitment to diversity, equity, and inclusion, it's incredible. It's something to witness in awe and to really, truly learn from. So it is a pleasure and an honor that I get to introduce one of my own personal heroes today, Dr. Gina Poe.
I am so excited to be here. This is my first keynote talk in person, and I'm just so thrilled. And it's been so great to get to know you guys over the last couple of days. I'm really sad that this is ending so soon. I would like another couple of days of this conference to talk to you all. But anyway, it's an honor to be here. I'm glad to be here where Black in Neuro started. And I'm just proud to be part of that organization. Yay. And yeah, it's, we already have a comment in the chat. Let's see, it looks like it. Should I go for it? Maybe somebody else can look at it because I can't actually switch the cursor to make it happen. Anyway, also here at UC Irvine is Sarah Mednick, and she just published a book, which is The Power of the Downstate. And so I took the title of this to, from her book, which is really, it is a super powerful time. And I'm hoping to convince you of that, all the power that can be wrested from the downstate. So you don't cheat yourself or your animals, research animals of that. All right, so let's get started. So this is the Fitbit image from my sister-in-law, Sandy, who's over 60 years old. And, I think she's got an incredible sleep pattern. This is very, very, very normal. She started out awake, and then she went into the N2 stage of sleep, and then into deep slow wave sleep, where you have these big slow waves that you can record from electrodes on the surface of the scalp. So, animals, any animal that has a organized cortex like ours, shows the same type of pattern. And then she went into the N2 state of sleep, where you have something called sleep spindles. They last about 1.5 seconds long, and they occur a couple of times every 10 seconds. And then she went into Rem sleep, rapid eye movement sleep, where you have beautiful theta in the hippocampus, the same kind of theta that you have during wakefulness when you're out and exploring and looking around the world and learning about the world. It's steady, it's huge. It's even bigger actually than waking theta. And you go back and forth through these cycles through the night. And in the first half of the night, you have this deep slow wave sleep. And in the second half of the night, you don't have hardly any of that. And it's mostly taken up by this REM and N2 stage of sleep. So that's the stages of sleep. So now what can sleep do for us? And does really memory require sleep? Well, we all homeostatically regulate it. If we don't get enough of it, we end up like this poor gentleman wedged on a train trying to catch a few Z's in transit. And so it really is required, even if we're not doing much, even if we're just kind of sitting around all day and rocking in a rocking chair and thinking, you still are required to sleep. But if we are learning, we need even more of it. So this is the outline I want to talk about. I won't have the chicken, so I am resting. He's sleeping here, but I still hope to get through in a timely fashion, except that I really want you all to interrupt me with any questions you have along the way, because it's a fairly intimate group here, and you guys are amazing. You've shown yourselves quite capable of great questions, and I don't want to ever lose you, and I want you to stay engaged. So please ask any questions as we go along. And of course, afterward, we'll have plenty as well. So we're going to go through the overview of the functions of sleep. When I started this in this field in 1988, there was very, very little known about the functions of sleep. We just knew we had to have it or we'd die. But we didn't know why. And now we have so much more. It's been really exciting to be part of the sleep research field over this time. And then we're going to talk about sleep across the estrus cycle, which is another area of actual, it's ground that hasn't been covered very much at all. So I'm hoping that some of you might even do some great research in this. And then the implications of these sleep across the estrus cycle and the functions of sleep for various things like mental health disorders. All right, so what are we doing? Well, you can think of waking as a big party, in your brain. So right now you're having a great party and your guests are bringing in information, which is gifts that they're giving you. And you have to figure out what to do with these gifts. And they're also eating food and they're creating a mess. But you're having a great time, hopefully. And that's expending energy. And you really have to wait until your guests are gone, consciousness is over, before you can really clean up. Because if you try and clean up during the party, well, then it's kind of the cleanup crew is weaving among the guests and it's chaos, right? So it doesn't work very efficiently. So you really need to go unconscious before you can clean up. And this was shown really excitingly just not too long ago in Niedergaard's lab, where she injected some tau protein into a mouse brain and then to see how fast it cleared. And in fact, during wakefulness, it hardly cleared at all. It took a long time. But if the mouse went to sleep, it was cleared within tens of minutes, actually, really, really fast. So that was super exciting. And that has all kinds of implications for the function of sleep, for preventing, for example, Alzheimer's disease. So we know that waking activity increases metabolites, and was said by someone up here earlier in the meeting. The beta amyloids and tau proteins increase normally during wakefulness, and sleep clears these metabolites through the lymphatic system. But then less efficient sleep really lessens the metabolite clearance. So what is it about sleep? Actually, when I was a graduate student, I was working with a colleague who had invented a new way to image the brain in these freely behaving cats. And it was just reflectance imaging. And what we realized, what we were measuring is the expansion and contraction of cell membranes as neurons fired. And that was based on some other data that showed actually whenever an action potential occurred, the membrane expanded. And especially happens out in the dendrites with those dendritic potentials, because the volume is very small. And so any kind of influx of water with the sodium ions really changes the volume. So if that's true, and neurons, when they're firing, they expand even a little bit, 15%. If during slow wave sleep, when those big slow waves are happening, all the neurons are, not all of them, but a good percentage of them are active at the peaks, and they're all silent at the troughs. And if that's true, then you get sort of a pumping action, right? You've got expansion and contraction and filling of the extracellular space. And also, it was shown by Giulio Tononi's lab that these slow waves sweep from front to back and sweep it into the lymphatic system. So it's really kind of a neat possible way that the brain can clear these metabolites that you don't want, just through the physical action of pumping. All right. So, and then one last bit, which is as we get older, the power of our slow waves, unfortunately, gets smaller and smaller. It's variable, depending on who you are and what your own slow wave pattern is, but it does tend to get smaller and smaller as we get older. So the efficiency of that pump would then also decrease. So the first affected area is that builds up these bad proteins is the area that's involved with attention, fear, stress, memory. It's the locus ceruleus in the brainstem. That's been shown as the first. The second, that it projects all over the brain, and it sort of brings those proteins down the axon to its targets, which are everything pretty much in front of it. The serotonergic system, the dopaminergic system, the cholinergic system, and then of course the cortex itself. So yeah, that's, okay. So disturbed sleep and smaller slow waves probably are the things that lead to many dementias. We know that they're correlated, and that disturbed sleep is one of the harbingers of dementia and other neurodegenerative disorders, actually. So it might be this. All right, so another thing we do during that deep slow wave sleep is we build proteins and repair ourselves. So this is the growth hormone release across sleep. And you can see in that first deep slow wave sleep period that you get a big surge of growth hormone. If you deprive yourself of sleep, you get growth hormone that ultimately ends up to be about the same, but it's distributed across the day. And we know, endocrinologists know, that there's a function to big pulses of things that's different than leaking it out over a long period of time. So this is an old paper by Carlisle Smith, and what he showed is that he injected some tritiated leucine amino acid, and he just measured the rate of incorporation of that radioactive amino acid. And you can see during waking and REM sleep, there's some incorporation. It's not zero, but during slow wave sleep, so proteins are really, really built in this state. That's different than waking and REM sleep. And that might have something to do with, I don't know, translation, probably. One of the things that happens during slow wave sleep is there's no acetylcholine, and we'll talk about that in a minute. So this growth hormone release is true in men as well as women. equally. So, all right. So another thing that happens in slow wave sleep is we are restocking our energy stores. So we all know mitochondria, the powerhouse of the cell, right? Well, it creates lots of ATP, which are the power packets of the cell. And so for transport to the various places it needs to go. And so what happens to ATP when it's being used for its various functions, it gets broken down to ADP, AMP, and finally just adenosine. And the amount of adenosine, as someone mentioned earlier, builds up the more hours you're awake. So you guys have probably a lot of it in your brain right now. So you've been awake for a little while, I'm sorry. So yeah, the longer you wake, the more adenosine. And we have receptors in our basal forebrain that senses how much adenosine we have, and it causes us to be sleepy. So it just says, hey, that's one of the signals that, hey, you need to go to sleep. And so, yeah, your battery is low. And so when you go to sleep, this is just 10-minute intervals in a rat brain. You can see the amount of adenosine gets incorporated back to ATP. So these mitochondria are really busy during slow wave sleep, converting that free adenosine back to ATP. And then when you wake up again, it just, you are losing that, building up that adenosine again over time. So just need to restock that energy store at night while you're asleep. Okay, so let's just talk. We've already talked a little bit about norepinephrine from the locus coeruleus and adenosine. So let's just go into some neurotransmitters and the milieu of the various sleep states. So during wakefulness, as you know, the cholinergic system, when you're paying attention like you are hopefully right now, your cholinergic system is really high. Your noradrenergic system is high, helping you learn, helping you to reorient if something were to happen right now. And the serotonergic system is also on. But when you go into slow wave sleep, one of the defining features of slow wave sleep that causes slow waves, enables slow waves, is the lack of acetylcholine. So that is really absent. If you measure with microdialysis or the activity of the cholinergic neurons, they're silent during this state. And in animals that have unihemispheric sleep, so like seagoing mammals, in order to stay at the surface and keep breathing, they need to have sleep one hemisphere at a time. And the hemisphere that's awake has lots of acetylcholine in it, and the hemisphere that's asleep has no acetylcholine in it. And that's the only thing that switches, the only neurotransmitter that switches between hemispheres. So it's a really cool thing. They also have a little less norepinephrine and serotonin. Those neurons are slowed down, but they are still active. And that's probably the thing that causes our slow waves, is that neurotransmitter milieu. If you give people acetylcholine antagonist, they will also show these beautiful slow waves. Yeah, even if they're sort of responding a little bit to you, it's kind of like a sleepwalker. They're able to respond, but they're not all there. So now in the transition to REM N2 stage, when I said we had this beautiful sleep spindles, really we don't have much of any of these neurotransmitters. This is another non-REM sleep state. And so there's still no acetylcholine, or very little. We actually don't know exactly how much, and it might be modularly, activated depending on what's going on. But we do know that if you have acetylcholine or norepinephrine or serotonin going on in the brain, the thalamus, which is the thing that allows these sleep spindles to occur, they're called thalamocortical spindles, that it becomes too depolarized and it can't produce spindles. So you really need an absence of these three neurotransmitters to be able to generate sleep spindles. So that's interesting. And I think we're going to talk a lot about sleep spindles in a minute. And then during REM sleep, tons of acetylcholine, even more in the hippocampus than you have during wakefulness. And that makes theta even bigger. And especially because you don't have norepinephrine, for some reason, norepinephrine and acetylcholine can sort of contend with one another in the amplitude of theta. So when norepinephrine is absent, your theta power is even bigger. And if theta has anything to do with learning and memory, and we think it does, then this might be a state when there's something really cool going on plasticity-wise. And so we'll talk about that. But we don't have norepinephrine and no serotonin that's coming from the dorsal raphe nucleus anyway. And I think that those things are, that neurochemical milieu is really important. You don't see this in wakefulness. And so REM sleep and non-REM sleep are both performing functions that can't be replaced by the waking state. Yeah. So, all right. So what's happening with these sleep spindles? Well, it's really cool because some papers by Siapas showed that spindles really provide a unique time when the hippocampus and the neocortex are uniquely connected. When the hippocampus fires, and this axis shows a gradation of more and more hippocampal neurons firing in a burst during non-REM sleep, what the neocortex does, and this is the prefrontal cortex, is it responds in a spindle frequency response. And in slow wave sleep before that, and in REM sleep after that state, there's very little connectivity, or at least back and forth between the hippocampus and the prefrontal cortex. It's really only during these sleep spindles when the cortex, is listening to the hippocampus. So this might be a unique time when memory consolidation is happening. The memories are being transferred out. So this is the spindle amplitude. Spindle is a 10 to 15 hertz frequency, and this is the amplitude at that frequency just after a hippocampal burst. And so sleep spindles are kind of cool because it was shown in about 2005. There was a sleep meeting, European Sleep Research Society, and I swear about a third of the meeting was all dedicated to spindles, because suddenly this is a state that people had ignored. It was so transient, so mixed, people didn't know what they were for, and so they just kind of ignored it, and they didn't even count it in a lot of studies. It was non-REM and REM, but the transition between them was kind of like, I don't know what that is, and so people didn't even count it in a lot of studies. But now we know that at least within a normal range of frequencies, that the time you spend in that stage 2, which is the N2 stage of spindle sleep, correlates really well with improvement across a nap. This is Nishida and Walker, but Sarah Mednick has also done really cool studies and continues to do cool studies with the function of spindles for consolidating memory. It correlates with IQ, and so suddenly in 2005 and thereafter, there's been a lot more literature than ever was before about sleep spindles and that state of sleep. So one of the really recent papers, cool findings, Julie Seept found, she found that when imaging the cortex layers 2 and 3, that every time there was a sleep spindle, and this dark blue is slow wave sleep, there was a ton of calcium that entered dendrites in the layer 2-3 of the cortex. So you can see during waking, there's hardly any calcium. I mean, there's some, but it's nothing like what you get each time there's a sleep spindle. Sigma activity is the sleep spindle frequency, and it's beautifully just up and down together. So this is layers 2-3. And so she also showed that if you recorded down here in the cell body, there really wasn't much going on. The cell wasn't really firing with each sleep spindle. Instead, it was really at these distal dendrites where all the action was happening. And so, this might have something to do with perhaps, the behavioral timescale synaptic plasticity that we learned about earlier today. It needs a big surge of calcium, and the timescale of it is not bad, actually. It happens 1.5 to 2 seconds. The longer spindles are, the more influx of calcium you've got. And then there's one other feature of this state, N2, which also has been fairly ignored because it's really hard to record these things. They're called PGO waves. The pontine geniculate occipitals, where they were found first in cats, they're big surges of glutamate, actually, that goes from the subcirrus, these glutamatergic cells beneath the locus coeruleus, all over the brain, well, first to the thalamus and then all over the brain. And they also target these distal dendrites. So it's the same state. That sleep spindles happen, and they are these big fluxes of excitatory activity that might be what these slow oscillations really are. Maybe the actual, it's hard in people, you can't really stick an electrode down in the subcirrus, so we don't really know, but we do have some recordings from the subcirrus in animals, and no one's done this correlation study. Are those slow oscillations really PGO waves? And maybe this is a state when there can be unique times of transfer and connectivity. Those distal dendrites are the cortical connections that are helping you update schema, for example. And this might really be the stage. This is Julie Seep and Christine. All right, so we did a study where we used dizipramine, which is an antidepressant that's hardly ever used, but it's a noradrenergic reuptake inhibitor. Or we used optogenetic stimulation to stimulate the locus coeruleus specifically during sleep. And we stimulated it at a rate that was low enough to not even disturb the sleep architecture at all. But it was enough to keep norepinephrine, it was 2 hertz, enough to keep norepinephrine out there at the synapses at a time when norepinephrine drops to nothing during those sleep spindle stages and during REM sleep. And what we found is that if we did a task where the rats were running around on a maze, and they'd already known where the food was, and we used Ensure. They love Ensure. It's sweet. It's milky. I don't know. Anyway, they will run for it. And if we used Ensure, they knew. And the other boxes all smell like Ensure because they've got Ensure in them, but they can't open the box and find Ensure inside. These are the only ones that they could open and find Ensure. And they pretty quickly learned to just run past these other unbaited boxes. But in a reversal learning task, what we did is we baited other boxes and left these unbaited that used to be baited before. So they have to remap this. And we leave all the cues in the room the same. So they actually have to incorporate this novel information into an old schema. They don't remap the whole thing, but they, as you know, they will form more place fields. Place fields will shift toward the goals. And what we found is that if you leave norepinephrine present during sleep, you will get an inability to incorporate this new information into the old schema. And what I already told you before is norepinephrine, like the other neurotransmitters, will prevent sleep spindles from happening. And so you count the transition to REM N2 state by sleep spindles. So you can't actually score transition to REM sleep when there aren't any sleep spindles, and the fewer sleep spindles they had, the less of that transition to REM sleep they had, the less they improved on this altered version of the track. And when we did it optogenetically, we also found that the number of errors went up with the change in that sleep spindle density. So the less sleep spindles they had, the more errors they did in this altered version of the task. These are animals that were controls, and they all increase their sleep spindle density, which normally happens when you're learning. All right, so the less transition to REM sleep and the fewer spindles, the less the new memory was integrated, and also we found the less the familiar memory was altered. So those sleep spindles are really important for updating our schema. So we've gone through clearing garbage, restoring our energy, building with proteins through these systems. We've gone into transition to REM, which is also called N2, where we are integrating new information into an old schema and consolidating, transferring from the hippocampus out. But what about REM sleep? Well, this is where I spent most of my own research, which is a REM sleep. And it's where we can really clean, I'm finding we can clean the synapses and sort of reshape our networks through this activity during this theta state. We can weaken old connections and strengthen new, kind of uniquely actually in REM sleep. So before I dive too deep into it, one of the unique properties is the locus coeruleus stops firing during REM, right? So here's the locus coeruleus in a rat brain, sagittal section, just projecting all over, starting from the front and ending in the back. Projects to the hippocampus and all of these areas. Here's the modularity, the possible modularity of these locus coeruleus neurons. A great series of papers by Dan Chandler. And it is silent only in that N2 stage and REM sleep. And it's silent for seconds before each of these sleep spindles, but then it fires and rapidly ends the spindle. So here's during REM sleep, some recordings done by my graduate student, Kevin Swift. You can see in that N2 state, transition to REM, also called intermediate sleep. It has a lot of names because it was ignored for so long, and every time someone rediscovered something cool about it, they renamed it. So just be aware of that when you read the literature. Yeah, so it's silent during REM sleep. So what norepinephrine does occupies beta receptors. One of the many things it does, occupies beta receptors, for example, in the hippocampus, which is rife with them. And it, when it's present, blocks depotentiation. So what would normally, this protein phosphatase, which would normally dephosphorylate all these things that are phosphorylated during long-term potentiation, can be dephosphorylated only when the beta adrenergic receptors are not occupied by norepinephrine. So yeah, so this is a unique time when depotentiation can happen. This is a review by Tom O'Dell in Learning and Memory in 2015. His lab was one of the first to discover that when norepinephrine is present, you can't get depotentiation. You can't reverse what was previously potentiated. So a graduate student in my lab currently, or Michelle, you want to raise your hand? Where are you? Oh, she doesn't want to raise her hand. Oh, there she is. There she is. Michelle is really looking into the mechanism by which we know what to depotentiate, what to throw out, and what to keep. So it's some cool experiments that she's doing, and I won't describe them in detail. I'll let her tell you the next time she gives a talk, but I'll tell you sort of the reason why she's doing these experiments is because we found that through doing literature searches that when norepinephrine is present, one of the things it does is it blocks and inhibits entorhinocortex layer 3 inputs through to the distal dendrites in CA1. So when norepinephrine is present during wakefulness, there's very little activity in this, what Olga Vinogradova called the familiarity encoding circuit. So that's during wakefulness. But of course, we know the novelty encoding circuits are quite active through the trisynaptic pathway, CA2 and CA3 to CA1. So a lot of activity. You can definitely get long-term potentiation in those circuits. But then when you go into REM sleep, you lose norepinephrine. So now activity through the entorhinocortex layer 3 can actually arrive. Also, when you lose serotonin, there are something called HCN channels that are also preferentially distributed out here at the distal dendrites. And serotonin causes a leak current to occur. And that means that when serotonin is not present, the leak current is not there. And so activity out here at the distal dendrites has a much greater chance of that dendritic spike, making it all the way to the axon hillock and causing an action potential. So, so you can see some firing that's a little different. And that combination of the distal dendritic spike arriving when actually it's an opposite phase of theta, when the novelty encoding inputs are not coming in, it could cause heterosynaptic depotentiation only when norepinephrine is not present. Okay, so the absence of those two neurotransmitters uniquely provide the opportunity then to depotentiate these novelty encoding circuits. So this is sort of the same thing I just said, but in another more complicated and mentally tiring graph. So I'll step you through it. So when something comes in novel and exciting and you're awake, and there's lots of norepinephrine, the locus coeruleus is firing bing, 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 and it's blocking this sort of familiarity encoding pathway here, the entorhinocortex layer 3, but not affecting the entorhinocortex layer 2. It actually doesn't affect that layer. And then the novelty can go in through its trisynaptic pathway. It comes in at the peaks of theta and can cause lovely long-term potentiation when this postsynaptic cell in the CA1 is most able to respond to that when they're at the peaks of that theta, ongoing theta. And then, but you're not going to get much activity at the troughs because this is pathway, which actually is an inverse structure. So theta at the proximal dendrites and theta at the distal dendrites are of opposite phases for some reason, but it's canonical. And so when the inputs come in through here, first of all, it's coming in at what's here, the theta trough. So you don't see a lot of firing at the troughs during waking novel learning, possibly because of that leak current too. All right. So then during N2 sleep, when the sleep spindles are targeting the distal dendrites here through those PGO waves and the spindles themselves, what you can get is beautiful LTP here out of the distal dendrites through the PGO waves. If you activate them alone, you can get LTP in distal dendrites. So all you need is that, but that plus sleep spindles, really guarantees that whatever comes in here can really be potentiated. There's low norepinephrine and low serotonin, but it's not gone, probably, perhaps. And especially at the beginning of the spindles, it is gone, but at the end of the spindles, locus coeruleus fires and ends the spindle. And so it might be that the beginning of the spindle and the end of the spindle does different things, but we also know the hippocampus is uniquely connected with the cortex. And that combined with spindles and PGO waves also allows the hippocampus to consolidate those memories out there in the cortex. And then during REM sleep, don't worry, I'm going to give you a mental break in just a second. So this is the last bit. So we can really refresh this novelty encoding circuit because now activity coming in through the entorhinal cortex layer 3 is no longer blocked by norepinephrine and can arrive strong and mighty. And PGO waves, actually the other time the PGO waves come in is during REM sleep, and they're actually active in bursts even more than during the N2 state. So really LTP can happen here. The cells can fire at what is locally recorded as theta troughs. And when these novelty encoding inputs are not arriving and cause that heterosynaptic depotentiation that can occur in the absence of norepinephrine. And the same thing might happen very well in the neocortex, although I've not looked into that so much, but I know that a lot of the same principles of organization are true in the pyramidal cells of the neocortex. All right, so how do we know what to throw out and recycle or what to keep after our waking party? How do we know whether we're doing long-term potentiation or depotentiation? And it might really do with the status of the consolidation status of that memory. So John Lissman came to UCLA when I was a graduate student there getting my PhD, and he gave a talk showing his new paper showing in a slice of hippocampus, when he added acetylcholine, you get this beautiful theta rhythm. And when he stimulated at the peaks of theta, and which I draw here because electrophysiologists like me are, kind of we record things upside down because we're outside the cell. But this is when the cell is most depolarized and most able to respond to inputs. You get beautiful long-term potentiation, even with just a single burst of 4 spikes at one theta peak. You can get LTP. And then you can get depotentiation, even more excitingly, when you stimulate at the troughs of theta. So if a cell is active at the troughs of its theta, you can get beautiful depotentiation in the slice. Is like a REM-like environment. You don't have serotonin, you haven't added norepinephrine. Those come from distal brainstem regions, and no one usually bothers to add it to their slice. So it's more like a REM sleep environment when you study the slice without adding those things. And you can get gorgeous depotentiation in this environment, which is super important, as you know, as perhaps we've talked about today, and you already know, to not oversaturating your hippocampus, right? You need, every time something's excited, you need to downscale something else. And I'm proposing to you that what you downscale is not done in a random fashion at all. It actually, during REM sleep, you are downscaling the memories that are already consolidated, and this is how it might happen. So when I went to the University of Arizona for my postdoc, here's a theta of a rat running around on a maze. These are the spikes of hippocampal cells, and you can see you get beautiful bursts of activity at the peaks of theta in a waking, running around animal, right? But during REM sleep, what I found is that those bursts switch to theta troughs in REM sleep. So I thought, oh my gosh, maybe REM sleep is really for clearing away memories. But the cool thing is the brain taught me something, which was that it cleared away memories probably once the memory has been consolidated. So a week into the exposure to an initially novel maze, that's when the cells fired at theta troughs during REM sleep. In the first couple of days, instead, they fired at theta peaks. So consistent with REM serving to continue to consolidate those memories during REM sleep. So REM sleep was initially for helping to strengthen memories that have not yet been consolidated to the cortex in the hippocampus. And then over the time course of consolidation, which other studies have shown takes about a week for a spatial memory anyway, that then after that period of time, you could switch to theta trough activity to depotentiate. So I want you to get up. Mental break, I promised you. So stand up. Those of you at home, if you can, stand up, or at least pretend to. I'm watching you. All right. And you can clap or snap and sort of move your legs, right? Because we're not only going to learn a jingle, but Autumn has taught us that we have to move too. In order to, and movement sets up this beautiful theta. So 1, 1,000, 2, 1,000, 3, 1,000. All right. So we have 4 hertz per second. All right. All right. And I'm going to sing it for you, and I want you to say it with me afterward. Okay. We're going to go 3 times. Ready? 1, 2. When acetylcholine's high and norethi's low, and the serotonin neurons firing real slow, and the forebrain's activated by PGO waves. These create the beat for LTP or LTD. Whoa, bidirectional plasticity. Whoa, reshaping network, storing memories away to learn efficiently. All right, you ready to do it with me? When acetylcholine's high and the norethi's low, and the serotonin neurons firing real slow, and the forebrain's activated by those PGO waves. These create the beat for LTP or LTD. Whoa. Bidirectional plasticity. Whoa. Reshaping network, storing memories away to learn efficiently. One more time, ready? Really got it. When acetylcholine's high and the norethi's low, and the serotonin neurons firing real slow, and the forebrain's activated by those PGO waves. These create the beat for LTP or LTD. Whoa. Bidirectional plasticity. Whoa. Reshaping network, storing memories away to learn efficiently. Yay! Give yourselves a hand. Yeah. And why did we do that? What? Yeah, we learned. All right. Advertisers know jingles work, right? So this is everything we've learned to this point. Good job. All right. Thank you. All right. So what about the hormonal cycle? Now the song goes from a ditty to a dirge because the hormonal cycle really affects all of these things. And so I'm about to tell you why and how. So this is the hormonal cycle in a human. You can see across the cycle, you got times when progesterone and estrogen are high, and times when estrogen is high and not progesterone, and so forth. And this is in the rat. Instead of over 4 weeks, it happens over 4 days approximately. And we go from estrus to metastrus to diestrus to proestrus to estrus again. So you can see during proestrus is when all of these hormones are the highest. And many studies have shown that rats learn differently at different phases of the estrus cycle. So how does it affect sleep? And this is actually, we heard a great talk yesterday about proestrus and fear learning, and it was kind of the opposite of everything I'm going to tell you. So I would love to know, the details of that study, because I'm sure they've got to be, that they have to coincide. It's just probably a matter of methodology. So, all right. So this is when women report the most insomnia and sleep disturbances, but there really hasn't been great methodological study, great method study with a lot of women. All these studies have been small, and although they're the best that could be done, they're just based on people's self-report of their sleep. But anyway, this is the time when women report the most insomnia and most sleep disturbances. And so we set out to see what happens in rats to their sleep across the estrus cycle. So these are some of this is graduate student Kevin Swift. These two are undergraduates, as were these two. And Karina is now a graduate student at UCLA in Ph.D. program, and Christy's in pharmacy program, and Yesenia is another graduate student in my lab who's taken the mantle of this work and really, really moving it forward in exciting new ways. All right, so this is in our hands, the measurement of the rat estrus cycle. You can see, well, this is actually from a textbook, but this is what happens to us too. We can actually measure the phases of the estrus cycle by swabbing the vagina of a rat really quickly and not too deep, so it doesn't cause pseudo pregnancy. And you can just roll it onto a slide. It's really easy to identify the stages of the estrus cycle. At least Yesenia, can do it really accurately. And you can see this is the proestrus phase, again, when all these hormones are really high. And then we took males and females and we implanted them with electrodes in the prefrontal cortex, over the CA1 area of the hippocampus, and the visual association area. And then we also got neck EMG so that we can really for sure know what stages of sleep they are in, because in REM sleep, you have atonia, so you don't act out your dreams. And so we can really tell. And then we have a commutator and we record for them for actually 5 or more continuous days. Actually, we have a couple of baseline days before this over the light dark cycle, 24/7. And you can see this is the light cycle and this is the dark cycle. ZT0 is 8 in the morning. That's when rats get their most sleep. And then this is the dark cycle when they get their most wakefulness. And then you can see where we did the vaginal or sham swab, and we did all the males, we did a sham swab because we wanted to disturb them equally. So then we just got sleep before and afterward all that time. And this is what a male's sleep looks like, really boring over day after day. It's the same thing, right? Okay, this is wakefulness. Yes, they're more awake during the dark cycle and less awake during the light cycle. This is the amount of non-REM sleep. This is the amount of REM sleep. And so they're asleep, 60% of the time during the light cycle. And as you might imagine, there's just not a lot of variation day to day. And then this is the sleep of females across the estrus cycle. And so what you might see is this time when women say they have the most insomnia, rats also have the most insomnia. Now, we don't know if they're bothered by it or not, but they are awake a lot more of the time during this phase, this proestrus phase. And the amount of REM, oh my goodness, very, very little REM sleep. And also very little non-REM sleep. There's a big dip here. The other days look, not so impressively different, at least, but that proestrus phase day is very interesting. So this is the amount of REM sleep, non-REM, and wakefulness in the proestrus phase, which is now encoded in red across the 24 hours of the cycle. All right. And yeah. All right, so and then green here is males. So you can see it's pretty much overlapping with males most of the time, except for that proestrus phase. Yeah, so this is just, again, once more, just isolating REM from the other two states, non-REM and wakefulness. And just, you can see there's a lot more waking and a lot less REM in the proestrus phase. All right. So what about the quality of sleep? So delta power, that's slow oscillations, those periods of silence and activity that probably sweeps, does many things, but it also sweeps the brain. Well, the power of delta is actually highest during that proestrus phase. So the power of that delta power in non-REM sleep is highest. Now they're getting less of it, right? Remember, they're more insomniac, but when they do get it, it's high quality, high powered delta. And then also true of slow gamma. I'm not sure what slow gamma means, but it has something to do with cognition. A lot of people, thinks it's something to do with cognition, and that's also, higher here. And I don't know if this is because, the brain, even when it's asleep, there's parts of it that are awake, and maybe that's why we feel like we have insomnia, because there's still part of our brain active. But in any case, it looks like it's pretty good quality, even when it's got high gamma. And then, so this is just the amplitude of slow waves here in the low frequency. And that time when the sleep is most disturbed. You can see it's just much bigger than the others. And if this is the low gamma, as you can see here. All right, in these various time bins. And so if you look at delta, you can see it here. And then you can also see there's some sigma. There's a little bit more sigma power also. So that's that spindle frequency that's going on. And then this is the slow gamma that you can see. And that's even more, and we see it during non-REM sleep, but even more in REM sleep. You can see there's more slow gamma. All right, so what about sleep spindles? Well, that is, this is what males look across like the 4 days of our recording. Sleep spindles are fairly similar across the period of time. But in females, it looks like the sleep spindles are actually lower in density per minute most of the cycle, except for the proestrus phase. And this is the sleep phase of proestrus. So when they're actually getting some non-REM sleep, the most non-REM sleep of that phase, the density of sleep spindles goes up. So again, even though it's not much sleep, it seems like it's, it's pretty good quality. And this is the coherence of spindles across 2 of our electrodes, the medial prefrontal cortex and the visual association area. And then during these three phases, it's actually not a significant correlation of spindles. So the spindles, when they do occur, are not highly coherent between brain areas, except for the proestrus phase. It's more like the males in that, when it's high in one area, it's high in the other. Spindles density is high in the other. We don't know what this means. We really don't. But spindles have something to do with something. And so this might be an important feature. And again, during that proestrus phase, it seems to be the most coherent sleep spindles. And we do know that, the number of sleep spindles goes up if you're napping and if you get lots of sleep spindles. This is a motor memory score in the no nap versus nap group. And those that had the most sleep spindles are the ones that did the best in this motor improvement task. So, all right, so how about theta? That was the last one we talked about, REM sleep theta. Well, it's a little less impressive, right? The increase in proestrus of theta, but it's still during proestrus that you get an increase in theta power when you're getting the least amount of REM sleep, but when you get it, your theta power is the biggest compared to all the others phases. And this is during the light part of the cycle when we're really not getting much REM sleep at all. And this is in different areas of the brain, the amount of theta that you can measure. All right. So, so how does this affect learning and memory? And this is something we're just starting to really get into. Yesenia is starting to do these tests in fear memory and especially memory that engenders in animals a PTSD-like phenotype. So one of the things that she's doing, she's moving from just measuring the amounts of sleep and these kind of fairly easy to measure things of the EEG, and she's actually putting electrodes down in the locus coeruleus to see how the locus coeruleus is active. Because in all the decades since the first person found that the locus coeruleus is inactive during REM sleep, that was in the early 1980s, no one's ever recorded from females. So yay, Yesenia is taking up the mantle and is trying to figure this out. And so this is the pattern that's been reported in males. And actually, so you give clonidine to make sure your electrodes are in the locus coeruleus because clonidine suppresses locus coeruleus activity. And so we can be sure that that's where we're recording from. You can see the clonidine really suppresses the activity, the firing rate of the locus coeruleus. And so this is what happens. In males, you get this beautiful pattern of step down and almost firing not at all during REM sleep. In females, at the low hormone phases of their estrus cycle, the locus coeruleus doesn't shut off, not nearly as much. It's at least twice as active as it is in males and maybe 2 or 3 times as active as females at the high hormonal phases. So what does this mean? Well, I gave you the dizipramine and the locus coeruleus stimulation. We're just stimulating at 2 hertz, and we get an animal that can't do a reversal learning task very well, can't incorporate new things into an old schema. Well, this might be a time when females also have, and that's three out of the 4 days of the estrus cycle, have more difficulty incorporating these new information into an old schema. Thankfully, that one day when sleep is super efficient and great, it's happening. But this might induce a lot of variability in studies of females if they don't control for the estrus cycle phase. So that's just a word of warning. So what about post-trauma? We just have a little bit of preliminary data, but this is the baseline data I just showed you. And then after trauma, a traumatic exposure, and this, we've tried 2 different ones. One's called single prolonged stress, which is the worst day of a rat's life, where you give them, bind them for 2 hours and then make them swim all together in a tub with no exit for 20 minutes. And then we put them in a bell jar with some ether, which also activates their HPA axis for just 5 minutes. And then we take them out and we isolate them for a week, where they can no longer socially commiserate with one another. It's the worst day followed by a really traumatic time, but it's actually a good way to produce a PTSD-like phenotype where they can't do reversal learning after this incubation period of a week. Or we've done it another way, which at UCLA is called, what's it called? What's that? Cephal, yes, thank you. Stress enhanced fear learning. So that's just 90 minutes in a box that has an electric grid floor, and randomly 15 times in that 90 minutes they get shocked. So there's no escape and they don't know when it's coming. And that's enough also to produce a stress enhanced fear learning and to cause a PTSD phenotype, weeks or months later. Anyway, post-trauma, you can see that during waking, right after the trauma, the locus coeruleus is firing a lot like it would because it's part of that stress axis. And then 4 days later, during wakefulness anyway, the locus coeruleus is normalized. But during sleep, although there's not that much change and it's not significantly different during slow wave sleep, you can see that after trauma, the locus coeruleus fires during REM sleep. And 4 days later, it's firing even more. And this is females, and this is at the high hormone phase. So when sleep is the most efficient and the best, you can see the locus coeruleus. Normally, it doesn't fire hardly at all, just like males during REM sleep, but after trauma, fires, and it fires even more. So what does this mean for reversal learning? For example, extinction of fear. Does that mean that really REM sleep can't do the depotentiation jobs that are probably important for incorporating new information like context-specific learning? And we don't even know yet what happens if you give this trauma at low hormonal phases. But we do know at lower hormonal phases, there's already more locus coeruleus activity at baseline. So we don't even know what yet might happen to locus coeruleus during REM sleep after trauma when exposed to it in a low hormonal phase. All right, so we did publish a paper during the pandemic, fairly early, with Yesenia and an undergraduate at the time, Jasmine Holloway, where we talked about what implications this could have. And so we just sort of built a conceptual model that if you have really good sleep before your trauma, your hippocampus, the novelty encoding pathways are refreshed, they're able to encode something new. And so you have good high associative memory capacity, and you can encode all the context and cues associated with that trauma, which helps you not generalize it, but rather keep that trauma specific to that time and place. If you have good sleep, and you have good sleep after the trauma, REM sleep with no locus coeruleus activity, then you have locus coeruleus, which is active during the time of the trauma, actually stops firing. And one of the things that actually helps the locus coeruleus stop firing in wakefulness before you even go to sleep is to learn something from that trauma. So if there's something to be learned, the hippocampus is there and ready to quickly cause LTP and all that, and that signal feeds back to the locus coeruleus to calm it down, so you can have REM sleep without locus coeruleus activity, which will help you then, set up those beautiful sleep spindles, because locus coeruleus is not active, not inhibiting spindles, consolidating to the cortex, all of that important information, and then allowing you to desaturate the hippocampus during REM sleep, so that next time you try and do an extinction or exposure therapy, your hippocampus is ready and to take it and able to, again, extinguish all that fear. But if you poor sleep before trauma, and you haven't had the chance to desaturate your hippocampus, like for example, if you're in the theater of war and you're not sleeping well at all, and you're stressed out, well, your hippocampus is probably not have as much time to desaturate, and you could have poor associative memory. So when a trauma hits you, you're unable to encode all of the complexity of the trauma and instead encode simple cue associations. And the locus coeruleus doesn't stop firing. REM sleep is, if you have REM sleep, especially early on, just after the trauma, when the locus coeruleus is still active, your REM sleep can't do the work it needs to do, and your spindles can't do the work it needs to do to consolidate all of the information to the cortex. Your hippocampus gets saturated and extinction therapy doesn't work. And instead you start to generalize and PTSD persists, and this sort of negative feedback circuit or positive feedback circuit continues. Your locus coeruleus continues to be overactive, at least during sleep. And so what can knock you from a good sleep to poor sleep, even if whatever happens before the trauma, is low estrogen, could cause the locus coeruleus to fire too much after the trauma, or if you have REM sleep before your locus coeruleus has calmed down. So those are the things that we're testing in the laboratory right now, but this is the model that we're working on. So I just want to thank my lab for the amazing work that they've done. Many of you are here. Raquel is here. And Ward is here. Paul is here. And several that, Sirana is here. Michelle's here. Yesenia, I think, is here. At least I know she was going to try. And if not, they're on Zoom, probably listening to this. So I want to thank all of you for making all of this research possible and all the things you're about to do. It's really making me excited. So yeah. Do not rob yourself of sleep. All right, thank you guys. All right, we have questions. I can probably somehow end the show, maybe in the show. I could start it again if people have questions that relate to this. Stop share? Yes, yes. All right. Hello, yes.
Thank you. I learned so much more. Can you talk a little bit more about the effects of sleep on young sleep and different types of sleep?
Yeah.
And which states of sleep they disrupt and what I imagine what issues have been arised.
Oh, that's a fantastic question. So unfortunately, there's been very little research on this, but I think it's extremely important. And the only poster I ever saw on this was about 10 years ago at a Society for Neuroscience meeting, or maybe it was a sleep meeting. Oh yeah, the question was, yeah, sorry, the microphone must not have been on. The question was, what does THC or CBD, anyway, cannabinoids, what do they do to sleep? And how might this affect all of this? And the one poster I saw was just incredible. What happened in rats, they gave a CB1 agonist. And what they found is that REM sleep was entirely replaced by constant 10 to 15 hertz sleep spindle. So they hadn't what looked like fairly normal slow wave sleep from what I can see on the poster. They haven't published this, and I'm just, that it was beautiful. REM sleep and the transition REM sleep was just one constant spindle, and it never ended. So I don't know. I really don't know what this would do to everything that I've been talking about, but the research needs to be done. The only human research that I've seen with, I think it was THC, was in a few normal healthy college men in Brazil, and it didn't seem to disturb the overall sleep structure, but they weren't looking into what was happening to the various frequencies of EEG. So I don't know, and it would be really cool to see what's happening. Yeah, thanks.
Thanks for the fabulous talk. I wanted to go back to the downscaling of the consolidated memories in particular. So if I'm understanding it right, would you interpret this as evidence against some of the like multiple trace theory? Or is that something that's part of like sharpening the consolidated memory?
So explain to me what multiple trace theory is. Is it?
Oh, so
Yeah.
Boris Moskowitz, Linda Dell, basically instead of the standard model where things are offloaded from the hippocampal trace and then removed and transferred to cortex, you maintain a hippocampal trace, but you have hippocampal independent.
Yeah, so I don't think it at all negates that because in fact, that potentiation at the distal dendrites would, in this model, at least at CA1, never be erased. So that would be kind of a permanent trace of the familiarity. It would be the entorhinal-cortical to hippocampal signature that this is familiar to me. And so it would be reactivated. During wakefulness, the trisynaptic circuit is still working beautifully. So it would still come in. And if there's any novel information, I'm sure that could get encoded during that state through the novelty encoding synapses. But what I think happens, at least, is during REM sleep, that gets refreshed every night when you're in REM sleep. So if it is familiar and consolidated, that novelty encoding circuitry anyway can be refreshed and restored and able. I think it works differently in CA3 because it's a whole different, organization of CA3. Entorhinal cortex layer 3 doesn't project directly to CA3 at all. So there might not be any theta trough activity in that state,
Great.
that area.
Thank you.
All right, thanks. Thank you for asking that. It's one of my favorite parts of the talk.
My question is, how do the sleep aids like the Z drugs like Ambien affect these frequency patterns, and how would you expect that to impact this effect with trauma?
That's a great question. So the person doing the best research in regards to sleep aids is actually Sarah Mednick, and she's actually paying attention to things like sleep spindles and learning. And there was a great study of schizophrenia that people with schizophrenia have very few, very weak, very short spindles. And it's probably due to there's a different calcium L-type channel in the thalamus of people with schizophrenia. At least there's a genetic association. And so those spindles are short and small. And so it might be that any sleep, and there was a great study that tried to enhance sleep spindles in people with schizophrenia by using one of these drugs. And they were able to enhance sleep spindles a little bit, and they were able to get better sleep consolidation of the memory a little bit. But I think that research needs to be expanded. The effect was fairly weak. And I think it needs to be expanded in terms of what all of these drugs are doing to all of these signatures. Well, the things that have been studied that the drug companies needed to get them FDA approved was just how much sleep, and not the quality of sleep, not the size of the slow waves, not the number of sleep spindles. And a lot of these sleep drugs actually eliminate sleep spindles or reduce them quite a bit. And a lot of them reduce REM a lot. So it's not necessarily, if you need or you want to prescribe a sleep aid, I think pay attention to these things we need to do more studies that actually look at all this. Yeah, thank you.
Thank you.
Thank you.
Hi, Dr. Poe. Great talk. So my question is, it's been shown that like most LC to CA1 projections are actually very dopaminergic instead of norepinephrine. And I'm curious, do you think that the high activity during REM in the trauma model could actually be due to like more dopamine release in the LC instead of norepinephrine?
Yeah, so it's not dopamine instead of norepinephrine. There's plenty of norepinephrine released, and it might be that dopamine is released under certain conditions like novelty or reward. But if the locus coeruleus is active, it's definitely, releasing norepinephrine. But you asked about, but dopamine and norepinephrine both work at G-coupled protein receptors and the same type of G-protein coupled receptors, and they both do the same thing to depotentiation. So they both block depotentiation and enhance LTP. So it might have something, there are different receptors for dopamine, just like there are for norepinephrine. So we do need to look at carefully at all of that, but yeah, I don't think it is all. Oh, so you asked if locus coeruleus is firing too much in REM sleep, is there also dopamine released? And I do not know the answer to that. That's a great question.
Thank you.
Thank you. Yeah.
Hi.
Hello.
Thank you so much for your talk. So you showed a striking result for consolidation over a novel maze where theta switches between the peaks and troughs. And that switch persists kind of over the multi-day time course of a consolidation. Yeah. I don't know if you have data to answer this, but could you speculate on, because within those 5 days period, surely the hippocampus is learning another memory, right? So would it spike up, or is it memory dependent? Is it a certain population?
Right, no, that's a great question. So if a neuron only encodes the familiar place, or at least from what we're recording, the familiar place, then it's firing at troughs if it's a familiar place. But if you expose the animal to a familiar environment and a novel environment in the same day, and some neurons fire in both places, some fire only in the novel, they only perform a place field in the novel, and some retain their place field only in the familiar environment. And what you get is a beautiful dichotomy. So two neurons sitting right next to each other, one that encodes the novel environment and one that's only encoding the familiar environment, will fire at opposite phases of theta. So it's just extremely elegant.
Maybe a follow-up. Do you have an idea of a mechanism?
Well, I think it has to do with the consolidation state, the state of potentiation at those distal dendrites. And I think once those distal dendrites have been potentiated, which is hard to do, but you can do in REM sleep with those and non-REM with those sleep spindles, then that sets up a time, especially during REM, or only during REM, when those distal dendrites come in, cause a dendritic spike, and cause heterosynaptic depotentiation at the novelty. So I think it's really dependent on the consolidation state, not only of the memory, but of that particular neuron's involvement in that memory. Thank you.
Quick question. Yeah. So I think I might be asking you to speculate a little bit, but you were showing that in females, they were getting like shorter bouts of sleep, but the quality of it was higher. Do you think that this, two-part question, do you think that this feature, the fact that it's like higher quality sleep, compensates enough for the like shorter bouts, or are these individuals sleep deprived, and therefore should we give menstruating people time off? Really?
Yeah, I think that's a great question, because it also relates to people with insomnia or short sleep cycles. And, is it possible that those who need 9 hours of sleep versus those who need 6 hours of sleep a night have, different quality of sleep, different efficiency of sleep? And we don't know the answer to that question, but I would like to think that it compensates, and especially because females, at least in our hands, learn better and able to consolidate better, whether they're learning the trauma or learning extinction of fear. If they're in the proestrus phase, they're able to consolidate that better. So I don't know about a day off. Might as well. Actually, I think menstruation happens the next phase during the low, when progesterone and estrogen are really low. So I would say, yeah, absolutely. Give them a day off for lots of reasons. There are maybe a couple of questions on.
There are probably questions for 3 days.
Oh, okay. I was just going to look online at the chat. Yeah, go ahead.
Maybe we should just do a couple from.
Yeah, let me see if there are.
And then we'll.
It looks like Sarah Mednick has raised her hand.
Oh, yay. Oh, okay. Sarah, you want to just unmute yourself? Yeah.
Hi. Can you hear me?
Yeah.
Oh, that's awesome. Oh, what a brilliant talk, Gina.
Thank you.
I was literally watching it while I was at my kids' swimming class. there's like 20 minutes I lost, and I just have a question for you.
Okay.
So the LC goes through tonic and phasic activation, right? So when you're doing, and the idea is that the brain kind of switches from this sort of stressed out state in tonic, and then goes into a more sort of calmer, able to consolidate, able to ruminate, able to, sort of process emotions and things, but in a sort of calmer way in that phasic state. And that's also where you see the lymphatic clearance. So what do you think is happening during REM when you see this LC activity? Would you categorize it as one phasic or tonic?
That's a great question. We've only recorded from a few animals. This is still very preliminary data, but so there's low tonic LC activity, which is actually calm when you're, writing something and you're at your computer or you're sipping a cup of coffee, but you're awake and you're alert. Your locus coeruleus is firing slowly but constantly. Then there's the high tonic, which is panic and it impairs cognition. And then for phasic also, there's times during sleep when it's not firing very much, and then times when it's bursting. And even during wakefulness, if it's low tonic, they can be, if something arrests your attention somewhere else, the locus coeruleus fires phasically and allows you to switch your attention to that other thing. So it's good for learning and memory, like you said. And then during REM sleep, what few recordings that we've done that I've seen is that when the locus coeruleus fires fast, the animal wakes up. And that's true of people with PTSD also. They wake up a lot more often out of REM sleep. And that's also true of people with insomnia. That's kind of the high anxious type of insomnia, is that they wake up a lot more. The REM sleep is more disturbed. So you could sort of infer, from that perhaps the locus coeruleus is firing during their REM sleep as well. So, and I think it's bursty, but I don't know for sure. And I think that's a great question. Anyway, I don't think it's high tonic because the firing rate overall is not, like the panic that you see in people right after a trauma, but it is too high for good cognition, at least in our hands. I mean, a good cognitive function later.
Okay, that's really interesting. Can I just say one more thing? Is that the guy who asked about what the Z drugs do to the spindles,
yes,
and then within that context of emotions. So we have one study where we looked at using the zolpidem to increase spindles, and then we looked at memory for a range of different type of emotions, from high arousal to low arousal, negative, neutral to positive. And we found that actually the increase in spindles with zolpidem also increased memory for high arousal negative images, meaning, that I think that's, obviously these are unhealthy people. And so there wasn't that sort of predisposition that to the negative experiences within anxiety or PTSD. But I think that there's something really interesting there that, if you take these drugs and you may have some sort of anxiety or PTSD, that they, if anything, the drugs may be, sort of consolidating the more PTSD-like parts of your memories.
Oh, wow. That adds another layer to it. Very cool.
It's sort of an interesting place to take that, to look at people with, with actual disorders would be really interesting.
Yeah, that would be great. Yeah. Well, I have just enjoyed all of your talks so much, and all of your posters that I made it to anyway. Thank you guys so much for inviting me. Thank you for being amazing and continue to be awesome. Thank you.